Good morning, students. This is 10th standard biology video class, video 1. In this video, we are going to start the new lesson, that is unit 20, breeding and biotechnology. Dear students, in this lesson, we are going to see what is green revolution, who is the father of green revolution in India, and also we are going to see the different breeding methods in plants as well as the animals. And in plants, we are going to increase the crop protection in agriculture. That also showing the different method. So that method also we are going to see in this lesson. Now, we are going to start this topic, green revolution. What is green revolution? Simply we can able to say that one, increasing the food yielding crop is said to be green revolution. So to increase the food yielding crops, we can able to apply the certain scientific technology methods or modern agricultural methods to develop the crop production in our country. Not only in our country, all over the world, we are going to increase the food protection by the application of modern agricultural technique under developed countries and developing countries. So, see the definition. Green revolution is the process of increasing food protection through high-yielding crop varieties and modern agricultural techniques in underdeveloped and developing nation. That is the definition for green revolution. Is an important 2-mark question. Simply, what is the meaning for that one? Increase the food yielding crop varieties by using the modern agricultural techniques. Dear students, Dr. Norman E. Borlaug is known as the American agronomist, is known as father of green revolution. Is in America, he is known as father of green revolution. For that, he received the Nobel Prize in 1970, Nobel Peace Prize in 1970. At the same time, in our country, Dr. M.S. Swaminathan is known as father of green revolution. He has introduced Mexican wheat varieties in our country. So this wheat variety increases the wheat production in our country and also increases the rice production in the year between 1960 and 2000. So this is said to be the green revolution. In this paragraph, we have seen 1-mark question. Dr. Norman E. Borlaug is known as father of green revolution. Dr. M.S. Swaminathan is known as father of green revolution in India. He has introduced Mexican wheat varieties in our country and also it increased the wheat variety and rice production in the year between 1960 and 2000. And also we have seen what is green revolution. The next topic is plant breeding for improved nutritional quality. So what is plant breeding? That means to cross the different varieties of plants to produce the newly formed individual with a different character or desirable character. And also we have to cultivate more protection in our country to increase the nutritional quality of the food material. That is said to be plant breeding for improved nutritional quality. Dear students, nutrition means taking food material in a side. At the same time, if you are taking less amount of protein content, which cause malnutrition in children. So to meet out this health problem, we have to produce the improved nutritional quality, variety of the crop protection. So it will increase the health of the human being as well as the animal. By feeding the high nutritional quality of the crops. So this nutritional quality is depend upon the quality and also quantity. Quantity means amount, large quantity of nutrients. So this nutritional quality is improved by different aspects. One is protein content and quality of the protein. So protein content is very much important of our body growth. So for that we have to increase the protein content food material and the quality of the protein. That means value of the protein. Next, increase the oil content of the food material. Then mineral content of the food material. So these three are very much important nutritional value to increase the health of a human being and animals. So that is said to be plant breeding for improved nutritional quality. Increase the protein content, oil content and mineral content. Dear students, how we can able to increase the nutritional quality of the food material? So one of the scientific process is known as biofortification. By using this method, we can able to develop the crop plants with the high level of desirable nutrients. That means required nutrients and also different variable nutrients like vitamins, proteins and minerals. That is said to be biofortification. It is a scientific process to develop crop plants with enriched food content with a high level of desirable nutrients like vitamins, proteins and minerals. So different examples of improved nutritional quality crops are given here. First one, Proteina, Shakti and Ratna are lysine rich maize hybrids. So lysine is an amino acid, that means a protein content. So these Proteina, Shakti and Ratna are the different varieties of newly formed maize crop with a rich lysine amino acid or protein content. These different varieties of wheat which are developed in our country, that is India. Dear students, see the diagram. This is the wheat variety named Proteina, which contains high protein content, that is lysine. Second one, Atlas 66, which contains the protein-rich wheat variety. The diagram is given here. Next one, another kind of rice variety is also produced. So we have seen two diagrams. Both the diagram is the high rich protein content of a wheat variety. One is the Proteina, which contains lysine. Another one is the Atlas 66, which contains the protein rich wheat variety. Next one, some rice variety which contains large quantity of iron. That is said to be iron rich fortified rice variety. And also some vegetables also produced with a high nutritional quantity as well as the quality. Example, carrot, pumpkin and spinach are which contains vitamin A, which increases the nutritional quality of the vegetables. So these are all different examples of plant breeding for improved nutritional quality by the process is known as biofortification. Dear students, next we are going to see methods of plant breeding for crop improvement. Dear students, the previous paragraph only we have seen what is biofortification is one of the scientific process to increase the high nutritional value of food material with the vitamin, protein and minerals, isn't it? So by increasing this nutritional quality, we are producing different crop varieties, isn't it? How we can able to produce those crop varieties? That methods are said to be plant breeding for crop improved method. So different plant breeding methods is there. Before going to see that one, what is plant breeding? So plant breeding is a branch of botany which deals with how to cultivate the different crop varieties with a different character and a high nutritional value and also increase the quantity of the crop variety, producing disease resistant variety. That means a plant which does not affected by any kind of diseases. Then insect resistant varieties. That means insect does not cause problem in the plant. That is said to be a branch known as plant breeding. In that plant breeding branch, we are going to see the different methods of crop improvement. First one, introduction of new varieties of plants. That means in our country, some of the food yielding variety of crops are not cultivated. Some crops are cultivated in other countries. So those new varieties are introduced to our country by plant breeding method. That is said to be introduction of new varieties of plants. Second one, selection. From a group of population, we are selecting the desirable character. That means a plant which are yielding high quality and also quantity of the crops is known as selection. Next one, polyploidy breeding. That means by increasing the chromosomal or doubling the chromosomal number in the laboratory to increase the crop variety with a variable character or newly formed character. Then mutation breeding. This is also sudden changes occur in the chromosome or by the application of physical or chemical substances to increase the crop variety is known as mutational breeding. Then hybridization. That means crossing of two different varieties with a different character. Both the parental characters are showing the different characters We have crossed those parents and we will get the newly formed character in the next generation. That process is known as hybridization. So first method, introduction of new varieties of plants. Dear students, it is one of the plant breeding process which introduce high yielding varieties of plants from one place to another. That's why I told you in some regions we are cultivating one kind of crops. That crop should be introduced to the other region or other place of its own country, or if you are introducing the different varieties from one country to another country, that is said to be introduction of new varieties of plants. So the species which are introduced from one place to another place, or one region to another region, or one country to another country is known as exotic species. Dear students, what are exotic species is the important 2-mark question. So these plant materials are, whenever we are transporting from one region to another region, these crops are carried the disease causing germs known as pathogen and pest. Pest means some kinds of worm, insects like that. So before introduce into one place to another place, we should test the plant thoroughly, and also we have to place the plant for separate region, segregation or quarantine. What is the meaning of quarantine? We all know that one. A person should be kept in a separate room for few days or few months. That isolation is known as, what is that one? Quarantine. Likewise, the plants with the pathogen or pest should be placed in the quarantine region before introduced into the agriculture field. So for example, Passeolus mungo, in Tamil it is known as pachaparupu or green gram, was introduced in our country from China. So this Passeolus mungo is said to be the exotic species of China and it is introduced to our country. So this is the one of the method of plant breeding, that is introduction of new varieties of plants. The second method is selection. So the selection is the oldest method of plant breeding. That means we have to place a group of plants with different characters. From that mixed population, we have to separate or segregate the highly desirable quality of a crops based on the external character or morphological character. That means with our eye we can able to identify the desired quality of a plant from the mixed population. That is known as selection. Dear students, this can be explained with a simple example. You are all the 10th standard students. Each and every classes, different students are there. That is said to be population. From that population, we can able to segregate the rank holders by external character. That means their activities. That is said to be selection. Likewise, from the group of a mixed population with different characters, we can able to select the desired quality of a plant by morphological characters is known as selection. This selection is divided into 3 types: mass selection, pure line selection and clonal selection. Dear students, what is selection? Write its type is the important 2-mark question. Dear students, see the type of your selection, that is mass selection. So we have to place here different characters with the seeds in a particular area, that is said to be a mixed population of a seed. From that mixed population of a seed, we can able to collect a desired character. Desired character means that means that plants which contain the newly formed characters. That is said to be desired characters. The seeds are collected from the farmers or from the any kind of the agricultural based departmental stores and cultivated in the agriculture field to produce the next generation. That is said to be the second generation. Likewise, this crop cultivation is taken place for 7 or 8 generation. At the end of the 8th generation, we can able to get a newly formed crop with a different character or quality. That seeds are selected and it should be given to the farmer and ask the farmers to cultivate the crops in the agricultural field to produce the newly formed variety of crops. That is said to be mass selection. That means from the mixed population, we are selecting the newly formed variety that should be produced for several years. How many years? 6 or 7 generation. Finally, at the end of the generation, 7th generation, the seeds which are produced with the desired character is given to the farmer for further cultivation. Some example of the mass selection crop varieties are given here. One is the groundnut variety like TMV2 and AK10 or the desired quality of a seed produced from mass selection method. Dear students, the mass selection method is given in the form of diagrammatic representation. See the first diagram, this is the first generation which are showing the different variety of seeds which are collected from the best plant. That is said to be mixed population. See yellow color, green color, blue color and red color is there. These are all the different seeds with variable characters placed in a particular region. It's said to be the mixed population. From that mixed population, we have selected the variable quality of a crop which is that is red in color, that is selected in the second generation, and also that seed is cultivated in a high quantity with a different quality or a character. Likewise, several generations we are doing the cultivation process. At the end of the 7 or 8th generation or 6 or 7th generation, we can able to get a newly formed seeds with a different character when compared to the previous generations. These seeds are given to the farmer for further cultivation to increase the nutritional value of the food material. That method is known as mass selection method. Dear students, next method is pure line selection. So in this method, we are going to select a newly formed crop variety by self-breeding. So what is self-breeding? See the definition. Pure line is the progeny. Progeny means newly formed species of a single individual. How many individual? Only one individual we are getting from the mixing of two parents or cross with two different parents obtained by self-breeding. That means we have already studied monohybrid cross and dihybrid cross. In monohybrid process, Mendel has chosen only one character with a different parent. Both the parent are crossed together. They have produced the F1 generation is known as progeny or newly formed generation. This newly formed generation is once again crossed with the same variety, that is said to be self-breeding. They are produced parental character as well as the desirable character of your crops. That type of selection is known as pure line selection. In this pure line selection is otherwise known as plant selection, individual plant selection. By using this pure line selection, we can able to produce large quantity of your crops. So these crops are selected from a self-pollinated crop. That means there is no cross-pollination. A self-pollination is taken place. So these crops are harvested individually and used for their nutritional value or food quality. Then after that, the newly formed progenies are evaluated separately. That means they will be taken into several experiments separately in the laboratory. Among those several individual progenies, we have to select the best one. The best quality or a crop is known as pure line quality or variety. So this pure line selection method, which are producing the progeny or a newly formed crop, which are showing the character is similar to the parental one in both genetical aspects as well as the external aspects. They are similar to the parental character. So whatever may be, the advantage of pure line selection is we can able to cultivate the crop in a large quantity. Large number of crops are cultivated by this selection method. But there is no newly formed variety because the next generation or a progeny which are produced by this method, which are showing the similar character of a parent in both genetical aspects as well as the external aspects. That is said to be genotypically and phenotypically. The next one, clonal selection. So this selection is otherwise known as a asexual reproductive method. Dear students, we already studied one of the asexual reproduction method is said to be vegetative propagation or vegetative reproduction. So here, a group of plants are produced from a single parent, not from two individual parents. Single parent, pure line selection means we have to cross the two different parental character. But clonal selection, we are producing large quantity of crops from a single parent plant through the method is known as vegetative or asexual mode of reproduction. Those newly formed plants are known as clones. So clones means, what is that one? Exact Xerox copy of a parental plant is known as clones. So according to this clonal selection, the newly formed plant is also showing the similar character of parental one, both genotype and phenotype. Sometimes we will select the desirable plant from the group of population. That selected plant from the mixed population is introduced into vegetative propagated method and cultivate large quantity of your crops. So this selection is said to be clonal selection. So from the mixed population, we are selecting one of the desirable quality of your crops, which is introduced into the vegetative reproduction. Those selection is known as clonal selection. So dear students, these are all the different methods of selection: mass selection, pure line selection, and clonal selection. Dear students, I hope have you all understood what is green revolution, how to introduce the newly formed plants from one place to another place. Dr. Norman E. Borlaug is known as father of green revolution. Dr. M. S. Swaminathan is known as father of green revolution in India. And also we have seen different newly formed crop varieties by producing the process is known as biofortification. Then what is selection? Then different types of selections are known as mass selection, pure line selection, and clonal selection. Then one of the crop which is introduced from China to our country is known as Phaseolus mungo. Dear students, up to this I conclude my video. Remaining we will see in the next video class. God bless you. All the best children.